This award is there to honor the first EMBL Director General. And it was launched in 2007 by Konrad Müller on behalf of the EMBL pensioners. And since 2010, it is sponsored by an EMBL friend and private philanthropist, Roland Specker. It consists of a cash prize of 10,000 euros and a medal. And it is given to a young researcher for outstanding contributions in science or science communication after leaving EMBL. So this year, the prize goes to Ilaria Piazza for her groundbreaking work on the global analysis of protein metabolite interactions in situ. And she will be introduced by Kiran Patel. Kiran, please, the stage is yours.
Okay, thank you, Fátima. My name is Kiran Patel, and I have the honor to introduce Illaria today, and I'm really happy for it. And I'm not going to take much of time away from what Illaria has to say, but just going to share a little bit about myself, just a tiny bit, and then introduce Illaria's work and put it into the context. I hope all of you can see the presentation now. Yeah, so this is the timeline of my academic life. Well, much of it is not interesting except this big box over here. When I worked at EMBL from 2010 to 20, I studied chemical engineer, and I was working mostly in biotechnological applications when I came to EMBL. And I think this is one of the strengths of EMBL that welcomed me in a way, an outsider to molecular biology, and really nurtured me and challenged me. And over the years, I have worked on many fantastic projects all around metabolic networks, looking into gut microbiota, looking into the nucleus, and many, many different fun projects, including that on wine and kefir and yogurts and cheese, etc. Another thing that I'm very proud of, that I have made a small contribution in starting the Green EMBL kind of a grassroots movement, and I'm really happy to see that the EMBL actually is contributing very significantly, both in terms of its day-to-day activities and in the new scientific program in making contribution, towards challenging the new global challenge that we're all facing on climate change. And that's really all enough about me. We're all here because of Illaria, and Illaria, many, many congratulations. I'm really proud and happy to see that someone working with metabolites has been given this award now. And Illaria she's now group leader at MDs in Berlin. She was EMBL Predoctoral Fellow in Christian Haering lab, and followed by a very successful postdoctoral period at ETH Zürich with Paola Pricotti. And she has developed mass spectrometry-based methods to analyze protein metabolite interactions in their native environment, and really global scale, not one metabolite protein at a time, but looking from a systems perspective. And this is, in my biased and still a global opinion, I think this is going to be a game changer in how I understand how cells work. And so, Illaria, many, many congratulations. And when I say that the metabolites are important, this actually I had to develop some thinking to convince initially to some of my colleagues. So when on the first day of my job, one of my colleagues, I'm not going to say names, asked me, why do you bother working with metabolites, and, and that made me think, and well, there are many different answers to this. First of all, metabolites, they make life, right? So primary metabolites, they really make our life, but equally important are secondary metabolites that make life worth living. And so we should really care about metabolism. The one issue in studying metabolism is that it's really complex and complicated thing, right? And so how do we kind of, disentangle this web complex thinking? What one needs to really do is to think outside box, right? Of course, conceptually, we need to put things in categories. We need to think about, nucleic acids, proteins, metabolites, central dogma, etc. But in reality, they're all interconnected. And in particular, the role of metabolites in regulating different cellular processes, not only at the enzyme level, but also at the level of translational machinery, transcriptional machinery, epigenetics, is really well understood. But in order to understand this in molecular detail, one really needs to think outside these traditional boxes. And this is what I think Illaria has done. She has, thought out of the protein box and metabolite box, but put them together in a network fashion. And so Illaria has been creative, and she has, really taken the courage to, go into this world of mass spectrometry, try to go into the field where you, really an interdisciplinary field that has a game-changing capability, and as she has illustrated successfully. So you need to be creative, you need to think outside box, you need to put a metabolite shoe and a protein shoe at the same time. And when you do that, the success is guaranteed. So congratulations, Illaria. And I think without more words, I pass it on to you. And I think now I don't know how we virtually do it, but I would virtually like to present you the John Kendrew Award.
Can you, Ilaria, hold up your medal so that everyone can see it?
Here it's coming out of the blue, the magic of Zoom.
Hey, bring it closer to the camera so everyone can see it. Hey, bravo.
Thank you very much. And thank you very much, Kiran, for the very personal introduction. I have to admit that I'm really moved also that you put these pictures with my friends. So it's great. Okay, so maybe I can now start my presentation. I tried to summarize a bit what I did in my short career. And I'm really happy that Kiran was giving a, very much metabolite-oriented perspective to it, because mine was, a bit different. So I hope now that I can summarize things really well. Can you see my screen now? Everything is fine? Okay, yes. Very good.
Fine.
So just a bit of arrangement. So first of all, I want to say good afternoon to everybody. I imagine that there are many people that are connected from different countries, even from different continents. So thank you for joining. And thank you for the EMBL faculty that is here today. Thank you, Kiran, again for the introduction, and for the Alumni Association for putting together this event today. I imagine this has been a colossal work to put together. And unfortunately, this is not happening live. And because from what I remember from my EMBL time, this was happening together with the EMBL summer party. That was generally, and I think it's still the highlight party of the year. So it's really a pity that it's not happening this year and also didn't happen last year. But I hope we can see each other live next year. And I'm really, really moved to receive this award that is now behind me. Because I'm succeeding a streak of great scientists that have already achieved so much, and also to be linked to the personality of John Kendrew that has done a lot for biochemistry and structural biology in general, that, as you will see soon, is very close to my heart soon as well. So okay, so I mean, this picture is summarizing also what Kiran showed you before. So I'm now a group leader at the Berlin Institute of Molecular Systems Biology and at BC in Berlin. But some years ago, I was really a very naive student that was doing an Erasmus project in Cambridge, UK. And by considering different places where I could do a PhD, I got to know the EMBL following a suggestion from a professor there. And after that, with this initial focus on biology, and then shift my interest on technologies, where I moved to ETH, as Kiran already anticipated. And then I had a short stint in the industry in a company in Zurich that was very interesting as well. So I had somehow 3 career phases until now. So how everything started? So everything started from getting interested in a very fascinating research topic that was, at the time, the biochemistry of mitosis. That's the reason why I joined the laboratory of Christian Haering in 2009, and in 2009, because I really want to understand how cells can fold chromatin into rod-like shaped structures that are symmetrically segregated at every cell division and are passed on without any trouble. And already there, it was very clear that there was one factor that is 5 subunit protein complexes that is called condensin that played a key role there. However, from a structural biology biochemistry point of view, not much was known about this protein complex because very scarce structural information was done before. And we really used to draw it like this, with this model that I have to say was quite imaginative because of these reasons. But what we knew for sure that this complex had these very two long coil-coil structures. And then this brought to the goal of my PhD project that was really to try to pursue structural studies of this protein in order to figure out how it worked mechanistically. And this brought me to, the goal of my PhD, and what this could be a graphical abstract of what was the aim. So I had these 5 proteins that were made of, very simple structural elements, so unstructured regions, solenoid repeated elements, in a complex that was too big for EM at the time, but was also not easy to crystallize. So I think, this was the typical project that was completely straightforward and easy, of course. And I imagine that the students are following the same kind of grant goals when they start their PhD. That's the way it is at EMBL. But then, I was young and naive, and so I just bring it on with me. And on the occasion of preparing this presentation, I really, went through my lab books. And I was, going page after page, and it really, my lab books really look like this, a lot of gels, insect cell expression, bacterial cell expression, a lot of limited proteolysis, trying to find constructs that could crystallize, and a lot of crystallization trials. And, I have to admit that this was not really successful, at least for the 2 years of the project. But this taught me a lot, and you can read the title here. That science is really not like in the textbook. It's never a linear story, and so you have to learn from these kind of difficulties. So you have to be resilient and also try to understand when it's the moment of shifting gears and do that. You have to do this in order to win a football match, but also to win over your PhD projects. So be resilient if you find now your time hard in your PhD project right now. But in fact, eventually, I think that you can find a solution for everything, as it happened to me, because we managed to narrow down a very interesting discovery. So, and that was that one of these subunits of the complex that is called YCG1 bound directly DNA. And this was not a typical DNA binding as we typically thought about that. So like the classical recognition of a transcription factor for a specific sequence, but rather a sort of a specific binding that was, functioning most like a docking event. And this was also, observed in vivo in chipset experiments, where we could see that our complex was really going into nucleosome depleted regions. And why was that important? Well, because with this, we could build a sort of first model of how condensing could work biochemically. And this taught us as the complex could be loaded and then potentially could, have this kind of looping extrusion activity. Of course, I simplified the story a lot today for the sake of time, but I learned 2 very important lessons from my PhD at EMBL scientifically. The first one is that every discovery then brings you to bigger things, because I was also very happy and proud to see how things developed in the following year after I joined, because in Christian's lab, but also in other labs, people have built on this, and they could indeed at the end solve crystal structure of the DNA protein complexes, and also with the great development that electron microscopy has had in the last years, also have complete models of the protein complex. So that was great and is still moving now today. And the second lesson that I learned was that by doing these genomic experiments with the Steinmetz lab, I could learn a completely different perspective, because in biochemistry, we're used to think in a very reductionistic way, So typically with the in vitro system, trying to simplify things a lot. And this is very powerful, but there is also an alternative way, that is the approach of systems biology, when you try to look at things in their global context. And then if you want to do this with proteins, the way to do that was to use a technique that is mass spectrometry, and this is still the way. And this is what drove me to visit the ground floor at the time where the lab of Martin Beck was. And I started to work a lot with this guy here in the picture that was a postdoc at the time, that is called Alessandro Ori, now he's also a PI. And we started to do some cross-linking mass spectrometry together that basically, confirmed that my protein complex was really flexible, and that was why it didn't crystallize. But, it taught me how, this structural proteomic technique was powerful to fill the gaps that we had in structural biology. And this, convinced me about the direction where I wanted to move for the next step. That's why I decided to go to Zurich. That was and still is an important center for the technological development of mass spectrometry. And I joined the lab of Paola Picotti for that reason. And this was my fast school for proteomics, unfortunately not for Swiss Deutsch, because it was really a hard task for me. My learning of German kind of worsened by moving to Switzerland. But then, that was, my motivation was really being inspired by the idea of expanding somehow the analysis of protein structures with NS technologies, and try to make structural biology feasible at the omics level. And somehow things came all together, because coincidentally, at the time, there were things cooking in Paola's lab. She started to think about this idea that then we further developed together, and also with other people in her lab, of this method that is called LIP-MS, that stands for limited proteolysis coupled with mass spectrometry, which essentially consists of the attempt to try to measure systematically protein structure variations in complex backgrounds. And again, this builds on something that I did in the past, so limited proteolysis experiments. So in these experiments, you try to figure out which regions of your proteins are flexible or are accessible in general by the action of a digestive action of a protease, by doing a digestion in a controlled way. And then use STS page as a method to visualize what are these regions that are differentially accessible. But then we thought that we could kind of extend this concept and do it not only for a protein or for a few proteins, but doing it for entire proteomes, and then use bottom-up mass spectrometry as a readout of this kind of phenomena. And then use mass spec, in fact, to build a proteolytic profile of whole proteomes, and use this as a proxy of protein shapes. So in practical terms, when then we try to visualize this, we would obtain some sort of structural barcodes for each protein in the proteomes, for those that are measurable, where we can visualize the regions that have increased accessibility or decreased accessibility to the action of the protease. So now you might wonder, why is this interesting to start with? Because it looks very abstract. But then, I would answer just by saying this general concept that, in general, protein can sense different environmental states of stress or conditions or perturbations in general by interaction events, and also by changing their biophysical properties. And this can trigger rapid adaptive responses that adjust the level of activation of signaling pathways and metabolic fluxes. So changes in protein structures can be used as a readout of protein activity, and also in general of cellular activity, especially when we are in a case where there is no change of protein levels. So in practical terms, what can we do with that? So we thought about using this for cover some gray areas in biology where there were not really tools to explore this kind of problems. And definitely one of these problems was to explore the relationship between metabolites and proteins, as Kiran nicely already explained to you. And in that sense, we tried to fill the gap by using LIP-MS to build a protein interactome of 20 metabolites of different chemistries and with different roles in biology. Initially using E. coli as a model system, because we intended to use this initially as a proof of principle case. And then what we figure out is that the panorama of metabolite binding in the proteome is much bigger than we thought, and that in general metabolite binding to proteins occurs beyond metabolism, but also can cover a broad range of cellular functions that go even into the direction of the cell cycle regulation. That is something that is completely unexpected. And of this, we could validate interaction that represented alternative substrate and products of reactions, and also have a regulatory role in the activity of proteins. Of course, this concerned the global map, the global picture, but remember that this is an analysis that is done at the peptide level. So with time, when we accumulated more and more data, we could realize that this, the position of these peptides that change in these proteolytic fingerprints in LEAP-MS is really informative also for the possible position of small molecule binding sites, because these peptides tend to be located in very close proximity to metabolites or drug binding sites. Thus, this LEAP-MS method has a structural resolution that goes beyond the identification of a single protein, and so it's powerful also in that sense. So far I've been talking about research in microbial proteins, which was very important at the beginning to establish this as something that could work. But then with time, we also developed it to make it work in human proteins that are, of course, much more complex than those of yeast or E. coli, for instance. And we started to work a bit more into the problem of the relationship between drugs and proteins, because these had clear implications for drug discovery and also translational medicine. And for this reason, we started also to interact with a biotech company that is called Biognosis. And just to give you an example, what you could do with this method is that when you have a particular drug that is bioactive, that, for example, could have an suppressive effect in the viability of your cells or your organs, often you don't know what is the molecular target of that molecule. And with this method, we can, just by adding a drug to a protein lysate, figure out what could be a candidate protein receptor, and also prioritize those cases that could be off-target effects or have lower priority, and those that are instead the most likely suspects. And at the personal level, this was also a very important step in my career, because I initially could start a collaboration with a biotech company in Zurich, Biognosis, first by just going there to their R&D lab and show them how these experiments could be done. But then I also later had a chance to work there for a short time. And this was a very interesting experience, I think, also for a person that has been rooted in academia for a very long time, like me, because there are lessons to be learned about how things to be done in a company. It's a very different perspective in general. So, and for what concerns what's boiling now, what's happening now, or what we have done more recently, well, with LEAP-MS, we can also explore protein structural changes that are associated with protein solubility transitions. So, for example, here you see, one barcode that shows the proteolytic profile of a protein that is called alpha-synuclein that can be present in a monomeric or in a fibrillar form. And this, of course, very important to distinguish this kind of two structural states of this protein, because this is a factor that is clearly involved in neurodegenerative diseases like Parkinson's disease. And this, of course, for the future has great potential for finding, structural biomarkers or something like this in biological fluids that could be potentially used for the early diagnosis of proteinopathies. And then again, going further ahead, another application that we could recently demonstrate a proof of principle for is that with this kind of structural proteomics, we can also follow, visualize somehow the activation of proteins in the scale of seconds or minutes or very fast cellular responses. And here you see a very, summarizing, perhaps even too much, cartoon, where you see the case of the activation of the osmotic stress responses in yeast. And basically, we could detect structural changes in proteins that are regulated allosterically or are involved in the increase in biosynthesis of glycerol or involved in the phosphorylation of the MAP kinase pathway that is activated when cells are osmotically stressed in yeast. And this brings me to, a sort of prospective slides, a sort of wrap-up slide of everything. So I started all of this by thinking, oh, I would really like to do structural biology with that kind of systems biology perspective. So can this be done? And what's next then in this field? Well, and this is also showing a bit where we want to go with my lab now and also in the future years. I think that technically we can now take different kinds of interactions and states of native structures that could be measured with the LIP-MS and also with structural proteomics in general, and try to integrate this information with classical high resolution structural studies. So for example, cryo-EM structural models of the cells that now are possible because we can really take snapshots of the cells, what's happening in a particular situation now with the great progress, the great strides that cryo-electron tomography has done. So in a way, if you look at things from the other perspective, we can think about annotating these micrographs with the data about dynamics that we can acquire with mass spectrometry based methods. And topic-wise for my lab, I really want to go back to my roots, going back to the EMBL inheritance. So we are trying to study how metabolite binding and in general metabolism can regulate gene expression and also chromatin structures. I really cannot say anything more because we are really short in time today, but I hope I could give you a good flavor of what I did in the past and what I want to do. And thank you again for the award. Thank you, Alumni Association. Thank you, Kiran, for the intro. And I have, many acknowledgements to say in addition. So first of all, I want to thank my lab that is still small but growing and ramping up with motivation. I, of course, want to thank my funding agencies that are supporting me right now or have supported me in the past. And this is probably instead the most important slide of the whole team here, because here I for sure couldn't list everybody and everything, but I hope I could at least mention all the categories. So people who have supported me, who support me currently, but the jury, EMBL, my former colleagues, the Steinmetz, Müller, Barabas, Beck Group, and everybody I interacted with, my scientific mentors, also during my postdoc, the industry partner, friends, family, and most of all, the EMBL pre-doc communities. I'm in contact with many of these people, not only from my year, but also other postdocs. And this, I think, is one of the best inheritance that you can have from EMBL, the network, not only from the scientific point of view, but very much also in the personal point of view. So thank you again.